Yeah. No, that's important stuff. Right.
Then they had these social reunions. I've never been on this campus. There are a lot of Illinois alumni.
Yeah, those guys are fun.
Yeah, there's a lot of them. It's funny, you're all here about the same name. So we can my director,
Contract. You can do it.
he was asked that level, you can pretty much. No. They do understand that.
Yeah, I do.
I was there. And I Maryland. Parents are from around.
Yeah, I can do it off the whole day.
Well, yeah.
Walking around, get a feel for it. It's been so dry.
Yeah, keep it.
Yeah.
I got rid of that. I got something that I want to grab. I mean, I saved stuff on my computer. I always save. I always have it there. So I just my computer for 5 hours. What? Now I got some extra prepared. Viewed that. IT set it up. Yeah. He forgot what he was supposed to put.
It's not that long.
It's not as wild if you get up in the tropics. It's we didn't even have to use the step up. Well, I was using the step up was like fun. I mean, even last year, I used the 2 months. There's still some fairly tall ones. Yeah, the last 2 years.
Yeah. Oh, I got it. I don't.
I like it. These buddies are pretty friendly. See how it goes.
Oh, bamboo greenhouse is basically everything all the time.
We appreciate your patience. We're actually just trying to track down Dr. Thompson and then he'll give his presentation. So hold on for 1 second, please.
It was just in here too. He probably went to the bathroom. Like gives everyone a chance to get down.
Seriously, Johnson.
Let me tell you something.
Short, the key. short sentences with key with A. Make it sound amazing. He was just here. I think he was there.
Yeah.
Probably Matthew spelled his name wrong.
It's like creation.
Yeah, he went home. Can't blame him. He hasn't seen these slides yet, but he's pretty confident. Wish he could wing it. Slides up, so. I'll go back to this. He's been looking great so far. All right, let's see if we go.
Good morning, everyone. Thank you so much for being here for this morning. And up next, I have the pleasure to introduce Dr. Steve Thompson. Dr. Thompson is a national program leader at the National Institute for Agriculture in Washington, D.C. Dr. Thompson received his PhD from the University of Florida in agricultural engineering. And prior to starting his current position at NEFA, he was a research lead scientist with the USDARs in Mississippi and a faculty member at Virginia Tech. His career has spanned various topics, from spray drift mitigation for aerial application for crop protection, to managing lots of projects related to either remote sensing, precision agriculture, and promotion of agriculture technologies to disabled farmers and also veterans. So if you could please join me in welcoming Dr. Thompson for his talk. Thank you.
Okay, I don't think I'll take the full 50 minutes, but I'm glad I have that long because I want to cover many different things. I tacked on the top of the title, USDA NIFA funding opportunities, because I think that's really what you want to hear. And then I'm going to talk about some of our new initiatives and things we've been involved with that are high tech and various things. I'm an engineer, but I do understand why you're so excited about genetics and plant breeding. I've worked with some plant breeders in the past on some other projects. So here we go. Okay, a little. Let me back up just a bit.
You have to point your blue plate.
There we go. Stephanie Pearl is my co-author. The reason is that I roped her into doing a presentation for the Agro Geoinformatics Conference in Fairfax, Virginia, and I ended up using a lot of the slides she used. So you'll be able to tell pretty quickly which are my slides and which are her slides. I'll just leave it at that. Okay, you can see this covers the bases on many things that we do at NIFA. We have all kinds of projects funded. That's basically it. A little update. Our budget for the AFRI program, that's our flagship program, came out of the administration as flat, which is quite good. There were a few initiatives that they took out, but the game is that Congress puts it back in. This happens every single time. Sunny Ramaswamy is continuing at NIFA. If you know about him, he will be on at least another year. Hopefully, we can keep him on longer than that. Sustainability is really what we're after. We have a new initiative, the SAS initiative, not the statistical package. It's sustainability something. I have it written down on my yellow pad, and I think I will get that pad. So prompt me on some of the stuff. You don't see it? All right, here we go. Sustainable ag systems. Well, I should have remembered that. Brain freeze, I guess. We have to treat this all as a system, okay? We are very interested in systems-oriented research. The SAS, the new SAS initiative, will cover the water management area that previously was set by itself, plus other areas. And this is an initiative, so I can't give you any details on what it's going to look like exactly, but you'll know pretty soon when the request for applications comes out. Okay. Leveraging the cooperative extension system is a big part. I'm going to talk about some of the projects that I lead. One that used to be a research-only project is now going to be integrated for the first time to include outreach and education also. And that allows us to bring in a few more interesting items. All right, I want to talk about how NIFA funds are provided first. This is NIFA 101. We have, it's pretty confusing. You know, if you look at these requests for applications, you can see, or you look at the NIFA website, there are a lot of programs there. And this is going to be an attempt to divide some of this out so you can get an idea. And I'm going to talk about some of the student-centered programs too. Many of the students can apply for these themselves. Faculty apply for most of these, but there are some that students can apply for fellowships and things like that. Capacity grants. This is formula funding that's given to the universities. Me and many of the national program leaders, we approve or send back or disapprove applications for these. And then, of course, the competitive grants. I want to mention one thing about capacity grants that I do to every university that I talk with, to the faculty. The quality of these things are quite variable. I will say that probably 80% of them are written very well. There's not a lot of quality control in some, however. I get these 2-3 sentence methods, you know, I can't, you know, that's going to be sent back. A couple of the NPLs will actually decline those. The problem is you have to start all over again. So I always defer in cases like that. So I'm, you know, when I came through, when I was at Virginia Tech, you know, I had a little committee and it was almost like a prelim exam. So it was pretty intense. So anyway, I just want to say that write those things up. You know, we do read them. I will admit that some program leaders are more stringent than others, but I'm one of those who, if it gets, you know, you're not going to get it by me. Anyway, all right, 30 competitive financial assistance program. The AFRI program is our flagship program. We have the SCRI, Specialty Crops Initiative. Tom Bewick runs that. Organic Agriculture, I think that's Matt Nagoju runs that one. Beginning Farmer and Rancher, a fairly new national program leaders involved with that. CPPM, I personally think some of this crop protection and pest management should be integrated a little bit better with what I do, the engineering, because we get some engineering related applications that are involved in this area. So I think we need to look in the future towards integrating that. And biotechnology, they call it the BRAG program. It's a great acronym I wish I came up with. And you know, this just shows the NIFA page where you can search for a funding opportunity. I'm going to mention a couple things here. These changes are mandated by Congress. Matching requirements that can be a burden on universities. Usually you can still be exempt from that. I won't get into the details there. Centers of Excellence has caused a lot of confusion. And there are basically 7 criteria for calling your grant. This is centered on the grant proposal. It's not like your universities, a center of excellence. But in writing up that part, it takes away from your page limit in writing the grant proposal. To give you an idea, this last panel that I ran, none of the proposals requested Centers of Excellence designation. All that's good for is if you have 2 proposals that are near the funding level or right at the funding level, and we have a limited amount of funds, the one that is a Center of Excellence will get the money and the other one won't. But this, we haven't seen, you know, this happen. Commodity Bourse provision is a good addition. I'm not that familiar with it, although I've had commodity grants when I was at the USDA ARS, and I think this is a good thing to single out, as they have. Okay, the organization of AFRI, we're aligned with the 6 Farm Bill priorities. We have, I'm going to talk about each of these areas. Foundational program, I run some of those. Then there are what are called challenge areas, and maybe not for the first time, but I'm going to attempt to tell you what the difference is between foundational and challenge. Those of you who are familiar with the NIFA and try to apply for grants. We have standard grants. CAP projects are huge, usually multi-institutional projects. Conference grants, okay, I'll give you an idea. Standard grants are about $500K within the engineering program. They can be $1 million or more in other programs. Conference grants are on the order maybe $50K, something like that. So if you have a good idea for a conference grant, it's a good idea to talk to a national program leader and ahead of time, because we're getting a lot of proposals in for these that are in the more high, you know, the high visibility areas. FACE grants, they're strengthening for underserved institutions, and then new investigators. All right, I'll talk about this one, the Eli thing first. Now this is where an individual can apply for a fellowship, postdoctoral fellowship or pre-doctoral fellowships. I don't remember who the program leader is. It may be Ray Alley who runs this. This is all on the website, though. I just want to make you familiar with these initiatives. Research and extension experiences for undergraduates. Okay, the institution applies for this. And also professional development for secondary school teachers. Same thing. Okay, here are the areas under AFRI. I'm going to center in a little bit on the one that's close to my heart because I have most of the programs. I'm the leader of this section. We have the agricultural engineering, bioprocess bioengineering, nanotechnology, and I'm also involved with 2 NSF programs. The last one is going to have many national program leaders involved. We had a new data science program leader position open, and it closed, and we are in the process of selecting someone who will probably be the lead national program leader for this. Okay, the NSF, we've hired a lot of we have not hired, we have funded these high throughput phenotyping projects in many different areas. Agricultural engineering has seen one. The National Robotics Initiative, we are funding some through that. The way it works with NSF is this. The applications come into NSF, and then they they dole them out according to the agency. Agriculture, we get some, NIH, DOE, various ones. And the ones that are in agriculture come to me, okay? The funding rate for both of these programs is pretty high. It's close to about 20%. We're getting typically 26 applications in, and there's 5 million in each program. So if you do the math, that's not bad. Ag engineering, bioprocess, we get a lot more, 70 to 80. Some programs get up, 100. Have to divide it into 2 panels. So cyber-physical systems deals with anything to do with sensing, modeling, adaptive control, machine learning, and then there has to be some type of control on the other side. National Robotics Initiative, we've seen some that involve
Do you mind standing just in front of the camera for the live stream?
in front of the camera.
Or in front of the microphone?
If you move in front of the phone.
Oh.
Yeah.
Oh, this thing isn't on?
No, now the camera is.
Yeah,
Oh,
exactly.
okay.
Don't worry about that.
All right, I'll stand here.
Perfect. Thank you.
I really wanted to be out of the picture, that's why. I think I may have turned that thing off too, so inadvertently. You'll find out. Anyway, we funded ones that have drones and ground robots working together, things like that. You can find the abstracts online if you check USDA NIFA CRIS. Google that or Bing that if you're a Microsoft person. Anyway, so just you can find out what's been funded, and the abstracts are there. Okay, food and agriculture, cyber informatics and tools. That's a new initiative that's coming out, and I can't tell you too much yet. These are 2 pretty cool programs. The CARE program is for critical needs. It's $300K each. That has had a high funding rate, except this year. We had a lot, the word is out, we had a lot of applicants for this program this year. I don't know if these are pre-screened, but I know the exploratory ones are pre-screened. This one is something I really like. This is for harebrained ideas. Okay, if you have a harebrained idea and 100K is enough for you, submit this thing. But it has to be really transformative. Some applications come in, they use the right keywords, but we know based on our expertise, and we can also look this up if it's truly an exploratory project. These are pre-screened by the national program leaders. So it'll come in, these will come into Charlotte Bear, and she'll send them out to the program leaders in various areas. I've seen many of these. And I'd say I've rejected about 50% that come in. Here's some challenge areas. I won't go through the whole list. All right, differences. Foundational program, mostly single function research. Okay, I said 500K, and they don't change that much from year to year. Challenge areas are mostly integrated programs. They are larger, and the research component is more applied. I did mention that my program, the agricultural engineering program, is now going to be integrated. However, we do have a few integrated programs within the foundational area. The FACE helps mainly underserved institutions. There is a list of FACE eligible institutions online, and you can see the whole list here. The seed sabbatical, I'm not as familiar with that. Equipment grants may be of use, of interest to some people here. New investigators, all the investigator does is just indicate that they are new investigators. There's no real advantage there except when you're determining whether the team has the expertise. Project director eligibility. Okay, for pre and postdoctoral, have to be a citizen or permanent resident. Okay, strengthening grants, EPSCOR. Don't ask me to tell you what that means, what that acronym stands for. I have the small to medium-sized institutions. I believe they have to be 17,500 or less. So if you're 17.6, I don't know. Minority serving is there's actually a flow chart online that a potential investigator can get, can answer these questions and figure out whether they are whether they are FACE eligible. The criteria for a new investigator: first 5 years in the career track position, limited publication record. I'm not sure what that means exactly, but and no previous federal funding other than pre- or postdoctoral fellowships. And this just kind of tells you what I already told you. It's a little chart indicating. This presentation will be on here for anybody to access. Oh, and I didn't mention this. These strengthening grants is at 11% set aside. So we will, in panel, we will rate the top proposals. But then, if there is a proposal that's from a strengthening state that is rated in the medium priority area, we will usually give the money to that person, to that institution. If it's rated not fundable, it's not going to get anywhere in any way. Higher education challenge areas. This is mainly for curriculum development. Okay, I want to break out the education and training part here. And we've already talked about the last part, but I want to center in especially on the National Needs Graduate Fellowships. All right, first, the multicultural scholars. I'm not that familiar with this program, but it's mainly for student experiential learning, and you can see the awards are about $200K. This one doesn't get enough press when we present this stuff. National Needs Fellowships. The reason I want to highlight this is because I was on one with the University of Florida. And it's nice because it gives the student a lot of flexibility. If they come in with an idea and they really have this, this is an example. May come in with an idea about something they want to do, and I'll just say it gives them a lot of flexibility to do, I will say whatever they want, but a lot more than, say, maybe an assistantship could give. So it's something for faculty to consider applying for. Here's a website if you want to get on the subscription service. This gives you updates on all kinds of NIFA activities. So I would encourage you to get on this. So, sure, this was fascinating. This is the extent of my genetics background. This is quite a genetic, amazing, heart on the left side, green blood, the whole thing, So anyway, okay, now I'll get back to issues, examples, and needs. Here we go. All right, you can see these are some statistics from the National Ag Statistics Service on the increase in yield in crops over the years, except for, I believe, non-irrigated in Texas. Let's not leave out the crops like cotton. We also have quality issues. How many of you have worked in cotton? Anybody here have worked in cotton? Okay. Well, you have cotton fiber quality. There are discounts for poor fiber quality, excess trash, things like this. So that's another index we need to consider when we're talking about crops, other crops. This gets into the remote sensing and precision ag arena. I remember when we started with precision ag, everybody thought that they could do wonders. Okay, there are some things that are not economically feasible. There are some that are, And so that's a topic for another discussion. And basically, I'm showing all of these. Let me see what I've written down. This is the genes, environment, climate management, and microbes, all working. You know, this is a system, and I like the fact that modeling is in this mix also. I don't know how many of you work with crop models, but you know, my feeling is using sensing and modeling together in a synergy would be a very good thing to continue. And I selfishly put this out because I used to work in it. Why do we use remote sensing? This is a simplified diagram indicating healthy plant versus stressed plant. There are many indices that could be used here. There's still a problem with transference of, you know, leaf level versus canopy level. You can work in the greenhouse and get some, you know, principal bands, but does it work in the field? Well, I don't know. There are new cameras out that exploit this area here, the red edge. If you have a stress plant, you're going to be offset slightly to the left, and you can see that by the slope of that line, it's quite a sensitive area. So I'd be interested to see how that plays out in use of these new technologies for, I think, mainly nutrient stress detection, the red edge response. We have a project funded by someone named Bruno Basso, it's a multifaceted project. We're dealing with spatial variability and also temporal variance, and I added that part, some observations on water management. We began to use, when I was with the ARS, began to use thermal imaging cameras for crop stress detection, crop water stress detection. That worked pretty well on a spatial basis. You know, a field, you could tell differences in stress and poor drainage. Using it for irrigation scheduling is another story, though, because you've got many different, you know, one day to the next you have different solar intensity, different wind, air temperature. Also, the altitude of acquisition makes a big difference in what the camera sees as a canopy temperature. So, you know, that's quite a challenge. They've had better success in arid areas with this because you can get a wider range of canopy temperature corresponding to stress, differences in stress. I'll leave it at that. And I already mentioned something about precision ag. Incomplete adoption, I have been to crop consultants meetings, or I went to one earlier this year, and one of the things they said is the speed of data acquisition to make decisions is a big deal for them. They want to want interpretation quickly from sensors. Many farm managers are using yield monitors, but then it's variable for the rest of the technologies. I said we have field and controlled environment phenotyping projects being funded. This is one, I needed my notes for this mainly. The one you see here was actually funded through the National Robotics Initiative. This is one I'm in charge of now. And basically reduces the non-productive time by acting as an intelligent courier service. So these, you know, it's using robots and humans together, and these trays come over to the human picker, I should say, and actually carry the product to the end. And it's just a scheduling thing with all these robots going down the rows, so the picker doesn't have to walk to the end of the field and deposit the crates and things like this. So it's pretty cool. This is out of Davis, California. We also have an SCRI grant, harvest aiding robots. Okay, the sensor goes through the handling process and measures mechanical stress on the blueberry itself. Eventually it will measure microbial contamination, and the outcome is better quality and less bruising, less waste also. So these are a couple of projects that are being funded that deal with robots. We have intelligent cutting and deboning. We just funded one out of the University of Maryland for crab processing that has some similarities, a robotic processing of crab meat. We also have aquaculture. I didn't mention that. That falls within our scope. Food waste is a big deal. Grant proposals that deal with that are going to be, and you know, this hunger map, I won't get in, there's still politics involved with the distribution chain on this one. I won't go any further than that one, but if we could possibly get past some of these issues. In the United States, we have, what do we call, food deserts. I'm slowly getting involved with the Smart Communities project with NSF. It's a possibility we may be able to interface our interests of urban agriculture, community agriculture, with National Science Foundation's goals also. Basically, it comes down to this: creating better transportation networks, supply chain logistics. Modeling is a big part of this. Also, the technologies and the acceptance issues is a big part. Social engineering, these kinds of social aspects. Data science. I mentioned we have a new data science position. And this person will be working with many of the disciplines that we're involved with. We have this cool package called PushGraph that allows us to get data like this, and you can see where the data-related research was from 2005 to 2008 as compared to now. Higher concentrations in, well, higher numbers in all of these areas, I should say. This is the new one coming out. Deals a lot with decision support systems and user-centered projects. And we have identified the needs and issues, or we've attempted to through our stakeholders, and we're pursuing opportunities for investment. This is new, so it's going to be interesting to see how this plays out. This first one, rewarding researchers to build off of one another's data sets, we'll see how that goes. Realistically. I'll let the rest of this speak for itself. Main points, okay, research that serves the end user is what it's all about. Serving the greater than 9 billion people requires solutions across the entire food chain, food supply and distribution chain. And we're interested in transdisciplinary research and solutions. And here's my email address. I have a limited number of business cards, and I hope this was of some use to you. Thanks so much.
Yeah, hi. If anyone has any questions, since we do have a little bit of time before the student speakers, we have some microphones. Otherwise, there will also be time at the roundtable discussion later. But if anyone has anything. But actually, for me personally, okay, especially with what you mentioned with like food deserts and all these different initiatives, I was curious as to how your average citizen would get involved in something like this without being directly involved in like the lab that's actually conducting the research. I don't know if that's something that you would know specifically.
Well, we have stakeholder groups you know, we also work with ARS, who has frequent USC ARS, who has frequent stakeholder meetings. So we try to center our programs based on feedback from that. As far as direct application, it's usually indirect after that. So.
Question. I mean, I can talk through that. So how when in your guys' decision-making process, obviously the end goal of the project is pretty important, but how about like the specific technologies that they're using? Do you guys take that into evaluation?
Yeah, our panel will. We select expert. You know, many in here have probably been on the panels. We don't have any specific, like our request for applications is pretty broad in the ag engineering program, for instance, and we just want to fund the best science. And so the experts who are reviewing these grant proposals will be able to determine whether these technologies are appropriate.
Are there any other questions?
All right.
Please think. Please
Thanks.
help me thinking.
I'm glad we're ahead.
Alright, we're going to start our student speaker session now. We're going to start with Austin Dobbles. He's a PhD student at the University of Minnesota under Aaron Lorenz. And he's going to be talking to us about applying drone technology to looking at iron deficiency in soybeans.
All right, can everyone hear me? That was more so to see if you guys were awake. No. All right, thank you for the introduction, Michelle. Thank you to the entire organizing committee and DuPont Pioneer for sponsoring this event. It's been a privilege to be a scholar recipient, to travel down here from the University of Minnesota. It's about a 7.5-hour drive across the beautiful states of Wisconsin and Illinois and Minnesota. So it's great to be here, and thank you again for having me. Also, shout out to all those people watching online. Thanks for tuning in. Shout out to my mom in there. I think she's watching. So I'm going to talk about iron deficiency chlorosis, and so before I do that, I want to define what iron deficiency chlorosis is to those of you that aren't familiar with it. And so what is soybean iron deficiency chlorosis? I refer to this as IDC throughout the talk, so I don't have to use all those big words all the time. So what it is, it's chlorosis due to a lack of iron in the soybean leaves, and the catch is that there's iron that's in the soil, but the iron is not in the available form that's needed by the plants. And so this deficiency occurs in soils with high pH. It also occurs in soils with large amounts of calcium carbonates. Those calcium carbonates serve as a buffer against pH changes, and then the iron is unavailable to the soybean plants, resulting in chlorosis. So I've mentioned this. Some of the symptoms of IDC include intraveinal chlorosis. You'll see yellowing of the soybean foliage, and you'll see stunting of the plant, as you can see here in this picture. So the problem with IDC is it causes tremendous yield losses. There's possibility of complete crop failure in some farmers' fields where they just don't get any return from what they plant. The entire crop fails due to the chlorosis. In addition, it's been estimated that losses annually can add up to over $260 million, so this is a great economic loss to the soybean profit as well. There are many solutions to overcoming IDC stress, application of iron chelates, various other strategies, management practices, but the very best solution to overcoming IDC stress is growing a soybean variety that is tolerant to the stress. So I want to show you here what this looks like, what a tolerant variety looks like. So I put a time-lapse camera out in my field, and on the right you'll see a tolerant variety. On the left you'll see a susceptible variety, and you can see that through time the susceptible variety is really struggling. It's not producing any leaves, whereas the one on the right is producing a nice big green healthy plant. So at the University of Minnesota, we want to go to our field and we want to select the ones that are healthy and big and green, and then the farmers, when they grow that in their field, they'll get a nice profit and yield from their products. So these are our control lines that we grow in our field, so we get an idea of how these lines are performing. But we really want to select those tolerant varieties. And in our Minnesota breeding program, in order to do that, we sort of have this funnel starting at crosses and advancing selected lines. But we do IDC screening on over 3,000 plots every year, everything from second yield trials all the way to regional trials. So we're testing a large amount of lines in our program for IDC. And this presents a problem in that we have a lot of plots to measure IDC from. So I want to introduce our IDC field site. So we have to find a location to grow these plots in which stress will occur. So I mentioned earlier that IDC occurs in high pH soil environments. So we have to find a location on a farmer's field or somewhere else that will have this high pH soil that's prone to the IDC stress. So over the years, research scientists at the University of Minnesota has adapted a site here in western Minnesota up near the Red River Valley, and this has high pH soil, and the farmer had noted it many years as having IDC stress, so it makes an ideal environment for us growing our trials. So I want to take you all to my field. So here we are in western Minnesota, the Google Maps. If we zoom into the satellite, this is what our field looks like. And if we grab the little street view guy and drop him in, this is our field out in western Minnesota. So this is what you'd see if you walked out there on a hot summer day in July. And if you pan around this view, you can see each of these plots are about 3 foot long, and we have over 3,000 of them in our field. So you can imagine it's nice to look at in the comfy air-conditioned room here or from your couch at home. But going to the field and measuring and taking notes on all of these is very time-consuming. So what we do is we go to our field, and traditionally what we do is we have either a pen and paper or we have tablets now, and we rate all of these plots on a score of 1 to 5. Scores of 1 are very tolerant to the stress, nice big green healthy plants, and the stress plants we rate 4s and 5s and completely dead down into the 5s. The problem with this, however, is it's subjective. It takes a very experienced rater to get good ratings that are consistent from year to year and from time point to time point. There's a lot of intra-rater variability, and it's very time-consuming, as I've mentioned several times. So the idea is we can use image processing to quantify IDC stress, and this has been successful in the literature. This is a study from Iowa State University and Danny Singh's group, and they took images from a tripod of hill plots and were able to successfully quantify IDC stress using various machine learning algorithms. My question when I started my PhD is, can we do something similar but from the sky, from UAV imagery? And so I'm going to talk about my research. Steve already mentioned this, but we're using the infrared spectrum. So here's the electromagnetic spectrum wavelengths on the x-axis, and on the y-axis we have the reflectance. And so what the human eye sees is we see everything in the red, green, and blue. But we can also use information from the near-infrared that really parses apart healthy from unhealthy vegetation. And so what you often see in the literature is the index known as the NDVI index, and that's just a ratio of the near-infrared to the red. And so we take a normal drone with a camera, we take off that camera, and we put on a new camera that is modified to capture information in the near-infrared. And then from that, we can calculate NDVI, and then we can fly this over our field and get data. So through my PhD, I've developed a pipeline, and I've also been using publicly available information. So I'll do an automated flight path in my field, capturing images with a 70% overlap. I'll orthomosaic all of those pictures into one composite field image using Pix4D. Calibrate my imaging using empirical line method. Basically, I have standards with known percent reflectance, so I can calibrate my imagery to this known percent reflectance. Then I can define where my plants are and pull out information from the near-infrared red, green, and blue of these plants, as well as how big the plants are. And then the idea is we can calibrate prediction models that go along with our ground-based data and then use that in the future to avoid the need for walking through the fields if we need to. So here's what image capture looks like. So we're flying at 200 feet, and the drone will fly a patterned route that's predefined. And so we'll capture with 70% overlap with the images, and that'll allow us to stitch our images together into an orthomosaic. So collected data in 2016 with just a regular Phantom without the near-infrared sensor, and noted very good correlations here. What I want to note is that we had 2 experienced raters rate the plots, and the correlation, the R-squared correlation between 2 raters was only 0.86. We had ground-based crop circle NDVI data, and that correlation was 0.76, so it's slightly lower, but still doing a very good job. And the UAV data was doing as good as our ground-based crop circle data. And so the idea is to improve the prediction accuracy and get better results as we move forward. So that brings us into data collection for 2017. The summer's winding down, things are starting to mature out there. So we did UAV imagery every week, and we also did ground-based visual scoring twice to build those prediction models. So here's orthomosaics from all the weeks. So here's early on, and then here's in July, and we see the plants growing. Well, some of them are really stunted, some of them are thriving. And so here's sort of a time lapse of all of the weeks of imagery. And then from that, I was mentioning we can put together NDVI maps. So this is the NDVI maps where green is high NDVI and red is low NDVI. And so we can see sort of where the problem areas are in our field. We can see where all of our plots are, and we can get an idea of where the stress is occurring. And then for this, I wanted to take information from these plots. Instead of just looking at it, I want to have the computer analyze this data. So I use QGIS to define my plots, and then I can pull out the average NDVI from each of my plots to get an idea of the stress of each of the plots. So this is the variation we're seeing in visual score data from the 0 to 5, 1 being tolerant and 5 being very stressed. So we're seeing a lot of scores in the 2s and 3s, but we do see a nice distribution of severity in our field. Pulling out just basic NDVI data from each of the plots and correlating that with our visual scores, we do see highly significant differences between each of our visual scores. So the 1s have a lot higher NDVI values, and the 5s basically have no NDVI. Just the plots were completely stunted. So we do see differences across each of our visual scores that we took on the ground from the UAV data. So I don't know how much time I have, but I didn't know how much time I'd have to go into all these details. But I think the benefits of this technology is to be able to look at IDC stress through time, something that wasn't possible before, simply because we could not get to our field site that was 2.5 hours away, spend 10 hours collecting data and coming back to the field. Back on campus. So I think this technology will allow a lot of information to be collected through time across all of these plots. Similar to that time-lapse camera, but across the entire field. Instead of just 2 plots, I can capture information across the whole field. In addition, part of my research is looking at a biparental population. So we took a highly tolerant line, crossed it to a highly susceptible line, and got over 500 recombinant inbred lines from that. And so we'll also be phenotyping this large population with drones and trying to correlate the genetic information with the phenotypic information that we're collecting from the drones to try to understand the genetics of iron deficiency chlorosis. In addition, this figure on the bottom right is an agronomic study. I'm collaborating with an agronomist at the University of Minnesota, Seth Nave. And so we created variations in IDC stress by applications of nitrates and by applications of iron chelates. So anywhere you see a blue box, that's where we added nitrogen. And so when we added the nitrates, we induced a lot more IDC stress, which you can see in this figure. All the plots, these are 4-row plots that had nitrates added to them, had a lot more chlorosis. In addition, we added iron chelates, and those iron chelates are highlighted in the green boxes. And those remove the IDC stress and allow for very thriving plots. And so we're going to be able to look at IDC stress of this through time and see if any of these plots overcome this stress. And we'll also be able to look and see how this correlates to yield components and various things like that. So that data hasn't been processed, but we did collect imagery every week. We collected ground-based data on this as well. And so we'll see how everything correlates at the end of the year. Here's my quick summary of what I talked about. We're developing methods for which UAVs can be used to phenotype IDC severity, and we're trying to improve our accuracy and efficiency of this process. A pipeline was successfully developed from image capture all the way to analysis, and now we can use that information to answer questions related to breeding, agronomy, genetics, etc. And so we can start answering these questions that weren't possible manually collecting data only 2 times in the year just due to logistics. So we're going to start answering more questions related to this stress, and I'm excited to see how the data turns out. This wouldn't be possible without making some acknowledgments. I'd like to thank my advisor, Dr. Aaron Lorenz. He's been a great mentor to me as I proceeded through my PhD, as well as some other members on my committee. I mentioned Seth Nave, collaborating with him, as well as some soil scientists and breeders and remote sensing specialists that have been a great help on my committee and give me guidance, and as well as research assistants on the crew. And I'd like to thank travel funding from the UIUC Symposium for allowing me to be here today. With that, I will take questions if you have any.
Does anyone have any questions for Austin?
Yeah.
I can talk about it. How do you separate genotypic response to iron,
Yeah.
presence or absence from, how do you separate that from variation in the microenvironment?
Yeah, so I'll repeat the question. So how do you separate the genotypic response from field variation? That's what you're asking. So that's a good question. We don't really know the best way of doing that, but I can say that we block our field in a way that we're only comparing, basically we have multiple tests in our field, and so we're only comparing to plots nearby, and we're not comparing from one side of the field to the other side of the field. So we're making selections from these smaller blocks within the field, which I didn't highlight due to time, but I guess another strategy would be repeated checks throughout the field to adjust, but we haven't done that just for logistic reasons. But there's various other ways to do it as well. Thanks for the question. Any other? Yeah, Taba.
Have you guys thought about going one level further into using satellite-based data?
So we asked about satellite-based data. Simply, the resolution, both spatially and temporally, of satellite data wouldn't be feasible for a breeding program. It's really hard to get less than 1 meter resolution from satellites. And also, we're limited to satellites can only fly, I think, like every 16 days would be another limitation as well. Whereas with drones, we can control when we fly, so we can capture those important time points of the stress, and we can get very good spatial resolution too. But for agronomic studies, or maybe for farmers' fields, wanting to make management decisions, I think that would be a good area of research. It's just not part of, it wouldn't be useful in a breeding program, I don't think. Yeah, I have 2 more questions. Wow. Good. No one was sleeping here.
This is a kind of an extension of the first question you got asked. So if we're talking about using this as a screening or agronomic tool in a farmer's field, so what we're detecting here is just a reduction in green biomass for the NDVI. And there are a lot of things that could potentially reduce green biomass that are not IDC. So do you guys have a strategy to try to pinpoint using spectral reflectors something that actually relates to iron efficiency specifically? And I guess also alongside that, what sort of environmental measures and controls would you try to include to separate that iron efficiency from specifically environmental or agronomic
Yeah. So that's a very good question, and it's a very challenging question to address. And I don't have the best answer, but what I can tell you right now is that what we're trying to do is improve our currently existing methods of hand phenotyping, basically. And so even when we're hand phenotyping, we don't really know if it's IDC stress or some other component. So we're capturing that imagery, so we don't know if it's IDC stress. I would like to mention that the agronomic study with the iron and the nitrogen applications, we also have another component to that with SCN stress. and we have variations in susceptible varieties. So those are two soil-borne pathogens, and a lot of time farmers will mix up, which is SCN, soybean cyst nematode, and iron deficiency. And so we are going to look at some of that, but we don't have any hyperspectral data at this point. But hopefully next year we'll have hyperspectral sensors so we can start to tease those apart more. But I will say that usually IDC stress occurs more in SCN-prone environments. So we might be able to see larger differences in yield than we'd see with just one stress over the other. Does that sort of answer your question? Thank you. Yeah.
Did you say anything about your ground sampling distance, your pixel resolution, or something about the resolution of the imagery that you do?
Yeah, so it's less than 2 centimeter ground sample distance, and that's a good point to mention. I do have ground control points for orthorectifying all the images so that from one week to the next they're spatially oriented. And so I use 9 ground control points in my field, and those stay out in the field all season long. And I also have radiometric calibration points. Yeah?
Do you ever leave some of the ground control points out to see what your resolution is using the ones that aren't used in your
So I have to clarify. So I have reflectance calibration panels, and I also have geometric calibration. Which ones are you referring to? The geometric. Yeah, I leave those out. They're just cement blocks, and so I leave those in the field all year long.
Do you use all of them all the time in your orthorectification?
Yes.
You don't leave any out and then recheck what your precision is?
Oh, no, I haven't done that, but that could be something that I could try. But if I have all of them, I'll probably use most of them. Thank you.
Are there any more questions for Austin?
I do have one more thing to say. So, I also want you guys to save the dates. So I'm a co-organizer of the Plant Science Symposium, but at Minnesota. So if you guys have smartphones and want to save the date, we will be offering travel grants. We have 2 symposia. The Production Agriculture Symposium is on Thursday, February 15th, 2018, and that will have a theme based on agriculture and an evolving climate. So agriculture dealing with climate change, and they'll have a lot of agriculturalists and growers and research extension people. So that's going to be a great event. In addition, our Plant Science, Plant Breeding Symposium has a theme centered around from markers to markets and breeding for end use quality. So if you have interest in those, or if you're a student and want to come present research, we'll have opportunities, so be on the lookout for travel grant opportunities and research poster presentations. Thank you.
All right, thank you, Austin. Our next student speaker is Chris Matthews. He's a PhD student under Dr. Lisa Ainsworth, and he's going to be talking about soy pigs.
Hey, can you guys all hear me? Yeah? Okay. Thank you, Michelle, and thank you all for giving me this opportunity to speak with you today. The title of my talk is Facing Climate Change. And so this is going to kind of go over the last 17 years of the soy-based research facility, what we've done, and where we're heading with this. So lunch is right around the corner. I should stand in front of the camera, as I'm told, and jump right into it. So we all know that the world is changing, right? The last 3 years have set records for global climate temperature, and the statistic is actually more alarming because 16 of the last 17 years have broke this record. And this has led to some pretty alarming events. The impenetrable doomsday vault in Svalbard flooded due to permafrost melting. Warmer Atlantic Ocean temperatures have set the stage for a horrible hurricane season, and we're still dealing with the aftermath of a lot of this. And the vast majority of this is being driven by carbon emissions, right? And so you can see here, is it a point? Can't point. Currently, CO2 concentrations are at 400 parts per million with annual increases of about 2 parts per million. I guess it's what's driving this increase in temperature, and the Intergovernmental Panel on Climate Change is predicting that we'll be at 600 parts per million by 2050. So in addition to causing increases in temperature, we as plant physiologists care about rising CO2 concentrations for a couple of reasons, one of which is that it's the primary substrate for rubisco. Right? So as we continue to see increases in CO2 concentrations, rubisco begins to favor the carboxylation event. As this occurs, when there's more CO2 at rubisco, stomates on leaf surfaces begin to close, so we see water use efficiencies. Specifically in C3 plants, and important agronomic ones, including soybeans, rice, and wheat, we see increases in photosynthesis due to elevated CO2. Biomass and ultimately yield is what we normally care about. With plants like C4, or with plants like corn, C4 plants, we don't really see these improvements in photosynthesis. This is just a carbon response curve. So, but if you look at C3 plants and soybeans, on the x-axis is our current atmospheric concentrations, and if you were to kind of follow the gray lines up and over, you see that there's opportunities for improvement. And on the opposite side of this, we have increased pollution due to climate change. Smog is coming to the United States now internationally, and local regulatory practices are potentially changing the current smog landscape in the United States. And why do we care about smog as plant biologists? This is because the primary, or the principal component of smog is ozone. And ozone is bad for plants as much as it is for us. Plants can't run away and go inside when it's a high ozone day. So they have to deal with this, right? So ozone will enter the leaf through the stomates, as you can see in this cross-section of the leaf. They'll enter the stomates, and they'll come into contact with cell walls, creating reactive oxygen species. This promotes cellular damage and decreases in photosynthesis. And these cellular level issues feed forward to leaf level and whole plant level responses. So more ozone decreases photosynthesis and reduces starch and sucrose production. It also causes decreases in total plant biomass, due to reductions in leaf area, increase in essence, and really importantly, decreases in yield. These two photos show really kind of the hallmark signs of ozone damage. The top photo is tobacco, one leaf grown in ambient ozone conditions, and the other one grown at an elevated ozone concentration. You see programmed cell death, that's the modeling, and in soybeans, you see chlorosis and bleaching due to elevated chronic ozone conditions. And so this is nothing new, right? Agriculture has always had to deal with variations in temperature and precipitation, diseases, pests, but changes in atmospheric concentrations of CO2 and ozone are a relatively new challenge and opportunity for plant breeders to take advantage of as the world is changing. And the only problem with this, it's really difficult to change the atmosphere in a way that we can measure things. And so in order to do this, we took it to the SoyFace Research Facility. SoyFace stands for Soybean Free Air Concentration Enrichment. It's a small 80-acre farm just south of campus here at the University of Illinois. And at this farm, we continue to maintain standard agronomic practices, right? So we do crop rotations, normal inputs. The only thing that we're changing is the climate in a small plot. We have 32 different rings, and we'll zoom in on one of these rings to see how they actually work. So each one of our rings are about 20 meters in diameter, and it has this green perforated pipe. So the pipe allows us to release either CO2 or ozone out across the canopy. Each one of our rings contains a small MET station that monitors wind speed and wind direction, as well as a sample cup for our gas of interest. All of this information is sent back to this control box here in a manifold. We rely on the wind to actually fumigate to carry this gas out across the rings. So as we release CO2, let's say, over the canopy, the MET station in the middle monitors wind speed and direction. If that changes, the controller automatically responds and opens up the manifold on the upwind side of the ring. This continues to do this for our set point, be that CO2 or ozone as well. We do a pretty good job of maintaining the set point or our concentrations that we would like to. The image on the left is simply CO2 concentrations in an elevated CO2 ring from a few years ago over the course of a day. And I wanted to include the thermal image on the right because, as I mentioned before, a C3 plant like soybean will close its stomates under an elevated CO2 environment. As you can see within the ring, we have these warmer colors indicating warmer leaf temperatures, especially compared to the plants that are grown at ambient just outside of the ring. So a lot of the work that has been done at SoyFace can be broken down into 3 phases. The first phase was just impacts, simply stated, how are plants going to respond to elevated CO2 and elevated ozone? Phase 2 are interactions and gradients. So let's add let's add another stressor to elevated CO2 or to elevated ozone. Something like drought or elevated temperatures. And the third phase, the one that we're currently in and that my research has really been centered on, is adaptation. How can we take the lessons that we've learned in phases 1 and 2 and apply them to make plants ready for a future a changing climate? So phase 1 was, I think, especially difficult for one reason. We had to take an 80-acre farm and turn it into a phase research facility. This required laying over 2 miles of pipe to deliver the CO2 and the ozone out to our rings. I guess we had to build a little town out there. We've got a couple dozen power poles and sheds and barns all across this 80-acre field. I can guarantee that the field crew hates planting SoyFace every year, but they come out and help us, so Jeff Warren, thank you for that. The map on the left shows the ring locations, so which treatments we're able to actually do in any given location on the farm. So the blue dots represent our elevated CO2 rings, the red dots are elevated ozone, the yellow, elevated ozone and CO2, and the green are our controls. During the early years of the project, the maize was always grown at elevated CO2. The ozone experiment was specific to soybean. We also had a combination of CO2 and ozone. And our target set point for the CO2 projects was always 550 parts per million. But with current trends and current IPCC predictions, we're going to surpass that by 2050. So our concentrations for CO2 have also increased. We're at 600 now for current projects that we're doing. With the ozone project at that time, our concentrations were always either 25 or 50% above ambient. And we simply measured ambient ozone out at the farm and increased the concentration within the ring appropriately. Phase 2 was primarily led by Drs. Leakey, Bernanke, and Ort. And they wanted to get at these interactions of CO2 and additional stressors. So Andrew Leakey's group needed a way to capture rain while still being inside a phase ring. There's not a lot of real estate in there, so they developed these drought awnings that would deploy in the middle of the night and then retract at sunrise. Luckily, in East Central Illinois, the vast majority of our rainfall happens at night, so during the 3 or 4 years of this project, they were able to capture 60 to 80% of the season-long rainfall. Carl and Don developed these heating arrays in order to answer these questions. What would an elevated CO2 environment as well as elevated temperatures do to soybeans and corn? These are season-long experiments as well as heat wave experiments. Phase 2 also allowed us, or gave us the opportunity to look at how a gradient of ozone would affect soybeans with different varieties. We grew a handful of soybeans and at a whole bunch of different concentrations of ozone, and it allowed us to get at any interspecific variation that might be capitalized in a breeding program in soybean. So phase 3 is where we're currently at, and this is all about adaptation. So what lessons we can take from phases 1 and 2 and put into crops to have them more ready for a future of elevated CO2 or additional ozone. So we're using transgenic approaches to do this. we want to create soybean plants that are more ready to better use the CO2. We're currently creating soybean plants that can handle elevated temperatures. And we're also using genomics and breeding populations to hopefully get at some of the mechanisms or the genetic underpinnings of ozone tolerance in both soybean and corn. So I would love to talk about all the great research that has happened out at this farm, but as you can see, there's not enough time to do that, ever. So I'm going to highlight a couple of key findings that helped drive my research over the last couple of years. So this is Kristen Bishop, and she was a graduate student in Lisa Ainsworth's lab. And all this graph is showing you, it's the yield response due to soybeans grown in elevated CO2. And we have two varieties that are pretty different in their responses. So Loda sees this huge increase in yield relative to ambient when grown in elevated CO2, and then you've got this other cultivar, HS93-4118, that, if anything, reduces in elevated CO2 concentrations. And so we wanted to take this, these results and see if we could kind of parse out why this is happening. Why are we seeing this interspecific variation in soybeans that are grown under elevated CO2? And so we stole Alvaro from Spain. He was a postdoc in Lisa's lab. And one of his many projects was kind of getting at why, right? And so in these panels here, you have seed yield, and they're correlated with 3 of the efficiencies that determine yield. The first panel is interception efficiency. The second panel is radiation use efficiency or photosynthetic efficiency. And then harvest index is on the side. And I highlighted the middle panel, which is photosynthetic efficiency, because that's where my talk is going and that's the research that I'm doing. But we also believe that it's a key determinant in yield. And on the flip side, Amy's research, another graduate student in Lisa's lab, former graduate student in Lisa's lab, saw that decreases in photosynthetic efficiencies were driving yield responses in elevated ozone. So on the y-axis, we have percent change. And on the x-axis, we have AOT40, which essentially is just a metric to see how much ozone is accumulated on a leaf over time. So on the left-hand side of that axis, we have our ambient, our control plots, and on the far right would have been the 200 part per billion plot. We have this linear decrease as ozone concentrations increase, which are being driven by reductions in interception efficiency and harvest index, but we see this pronounced decrease in photosynthetic efficiency. And more recently, Tim and Nicole, Tim is here, thank you, Tim. We're seeing interspecific variation in maize responses as well to ozone. This plot shows 45 different hybrid cultivars, and our average reduction is about 15% in photosynthesis. But what we're really intrigued by and excited about is these differences. Some of the varieties don't change at all, and some have these really pronounced decreases in photosynthesis. And so we believe that this research shows us that photosynthesis is a key trait in describing plant responses to atmospheric change. The only problem with measuring photosynthesis is it's really difficult and it takes a lot of time. I'm going to pick on Tim because I can see him right now. But if you ask Tim how easy it is to take photosynthetic measurements on 45 cultivars that's fully replicated 4 or 5 times out in the field over the course of a season, he starts getting cranky, right? So this is where the vast majority of my research comes in. Can we develop a method to quickly measure photosynthetic parameters or photosynthesis so that these smiles stay smiles out in the field throughout the course of the field season? And so again, that was the goal. Can we develop a high throughput phenotyping tool to measure photosynthesis? The traditional way of getting at photosynthetic parameters are with the CO2 response curves that I showed you earlier, kind of botched my explanation of, but one of these curves takes about 30 to 40 minutes to do correctly. And during this time, you're getting a measurement on one plant. Using hyperspectral reflectance measurements, especially the full spectrum, with something like a ASD field spec or any sort of spectral radiometer, I can get measurements on about 100 plants in the same amount of time. If we have the ability to build models that can accurately predict, these photosynthetic parameters, or whatever your gold standard trait is, could really move things along quickly. And so, in short, we did it. We're pretty excited about the top 4 panels. Using partially squared regression modeling and these hyperspectral reflectance measurements, we were able to build predictive models for chlorophyll, nitrogen content, specific leaf area, and I have Vmax. It's just the maximum rate of photosynthesis for these corn plants. I really wanted to include the bottom panel because it doesn't always work. All right, this isn't magic. It is a mathematical model used to build these predictions. And I really do want to reiterate that this reflectance data is really special in that it takes me 30 seconds to get a measurement of a plant. And depending on what indices you want to use, and DVI was spoken about earlier, you know, I've got 4 or 5 really difficult traits out of one measurement. And so we brought it to the field, right? We built this brand new model to get photosynthesis measurements that are really quick. There's no more wet chemistry. We're not grinding tissue samples. And you know, from 2013 to 2015, we grew a whole host of different inbred and hybrid maize populations under ambient and elevated ozone. And our models are working. They're pulling out differences not only between our treatments, but between the populations that we grew. And I guess that's the real take-home that I want to show from this. But I also want to show that, I mean, this kind of looks similar to the work that Tim and Nicole did. The model and then using hyperspectral reflectance measurements are pulling out the same sort of interspecific variability in these maize plants grown under elevated ozone. I'm not trying to put Tim out of a job, but you know, we're excited that we're seeing a similar correlation, right? And we're currently working on writing this up at the moment. And so we're not done there. We also have grown 2 different mapping populations, near isogenic line populations under elevated ozone. And we're planning on using these tools to see if we can pull out QTLs for ozone tolerance. We've also developed a half diallele population, which are those 45 hybrids that you see these measurements on, to see if any of the traits that we are currently seeing or excited about in these projects are actually heritable, right? If it's not heritable, you can't breed for it, kind of defeats the purpose. I've been sitting on a lot of this data because I've been a technician as well as a graduate student and a TA over the last couple of years, but I have the same sort of data with soybeans. We grew an RAL population a couple years ago in elevated ozone, and if they're successful, and I really hope that they are, because it's my PhD project, we're gonna apply those to that population. And I was lucky enough to work with Carol Bobbin, Carol Fox. Some of you might know her. She was in Brian Deere's lab, and she took the field spec backpack and took leaf reflectance measurements all across Nebraska and Illinois on the soybean NAM population. So our biparental population is great, but you get a lot more power using an association mapping panel, right? And so I'm really excited for this winter to walk away from the farm for a little bit and get this going. And photosynthesis is also a tool that is a trait that we're not usually looking for in germplasm screens, right? I think part of it is because it's difficult to measure. It takes a long time. So this can be a new tool in a plant breeder's toolbox as they move forward. I'm looking for ways to adapt crops to a changing world. And really importantly, we like to see if this data will actually scale up, if our models will work. Currently, these measurements are being done at the leaf level. We're working with Carl Bernanke's group, he'll be talking later, about trying to get these models that we're building at the leaf level to something on like an autonomous drone, on a push cart, anything to speed up this process both in time and in space. And so, I guess in conclusion, we wouldn't be able to answer a lot of these questions without the Soyface Research Facility. The ability to maintain a farm in a normal agronomic practice and just make these slight perturbations to the climate is really unique. As far as I know, we are still the only FACE site that's running in the US. And we're not that big. Again, it's an 88-acre farm and our rings aren't super huge, but we do have the ability to take advantage of some of the genomic resources to help answer some of these questions. And in order to continue pushing our research forward, we've been coming up with new innovations. Drought awnings, infrared heaters, and now building hyperspectral models that are seemingly high throughput. I'm currently carrying the backpack still, but at some point we'd like to get it onto a drone for a changing world. So I've been lucky enough to be able to work at SoyFace as a technician, as a graduate student, and as a graduate student research technician for Lisa Ainsworth. But SoyFace wouldn't be here and wouldn't be such a great asset, and we wouldn't have had all those great publications come from it if it hadn't been for Evan DeLucia, Steve Long, and Don Ort. They made sure that it happened, so thank all of them for that. Andrew Leakey and Carl have been great support with my research and run a lot of the projects that are happening at the farm at the moment. And Lisa Ainsworth for taking a risk on an international studies student and giving me a job and then school. So finally, the technicians that help me run the farm on a day-to-day basis. Aiden, whose last name I misspelled, Jesse McGrath, and Tim Meese for helping develop and build SoyFace from the beginning. And the high throughput phenotyping project, my cohort, my guys were Craig Yendrick and Tiago Tomez, and then all of the labs that work out at the farm. So with that, thank you. Questions? If you guys want to come, you can still see the stuff in action too. Andy?
Yeah,
absolutely.
On the hyperspectral data, if you could find significant wave bands and then only build sensors with only those wave bands specifically.
So the models that we're currently building, we're avoiding that because we do have the full spectrum to work with. Right, the sensor goes from 350 to 2,500 nanometers. You in theory could, right, and then you could build a smaller sensor, and there's not. I think mathematically it kind of breaks the models that we've built. So in theory, yes, you could start to see which are the most significant wave bands and see if you can still get really decent predictions with them. But this technology is improving and getting so much faster and smaller. I mean, the idea of having a hyperspectral camera that's this big in like 5 or 10 years that can go on a drone, I think it's better to keep the stats legitimate at the time. But if you're just looking for something like NDVI, you've got it right there, right? So that might not be answering your question, but yeah. That it? No. Hi, Jack. Yeah.
So what you indicated that you are actually been working with transformation
Yeah.
in terms of soybeans.
Correct.
How did you identify candidate genes?
So what they're actually doing is I don't know if Pauline's here. This is primarily her work, but photosynthesis tends to see a reduction in elevated CO2, at least the VC max, right? And so they're trying to find a way to stop those reductions in photosynthesis, and they're adding like novel transporters, sucrose transporters, to help shuttle sugars out of the cell and into different parts. A lot of research has been done with this on changing like the sink source ratio and the size of like soybean pods, if that helps continue to promote increased photosynthesis. So that is one of the ways that they're doing it. I don't think it was so much a candidate gene in a traditional breeding sense. Yeah.
Can you say, I was wondering about the models that you developed with the hyperspectral data. Were those in the greenhouse? Is that where they were?
No, so I know it was a noisy plot, and so I'll just go back to it real quick. Yeah, so panel A would have been the model that was just built based on outdoor measurements. So we collected all the different genotypes that we planted in both elevated and ambient ozone during the 3 years of the project, at least that part of the project. We also did a greenhouse experiment where we gave them low and high nitrogen to tease out if we were just getting nitrogen hits, right? I don't show the correlations between the different traits that we're modeling because you might just think that we're picking up greenness, right? Yeah, and so the panel on the right, it doesn't really change the predictive ability of the model if we include the low and the high nitrogen plants. So that's the difference between models A and B. And so we really were cognizant of that when we started the project. It's like, if this is just a giant SPAD meter, we're not getting real measurements of photosynthetic capacity, then we really don't want to continue to use it. Because it's expensive too, right? At the moment, they're like $60,000. Yep. Lunch? Cool.
Great. We'd like to thank all of our morning speakers and invite you up to lunch upstairs and for the second poster session. Thank you.
Thank you, Jeff.